Everyone, welcome to the Biodecoder channel. In this very quick version of PCA, I will show you how to analyze wheat trait data using principal component analysis in R. So my data set has 4 quantitative traits: plant height, grain yield, 1000 seed weight, and protein content for different wheat varieties. There is also a qualitative variable treatment which shows whether the plants are controlled or fertilized. I will use PCA to see which trait and varieties contribute the most to the variation. I first select the quantitative traits and run the PCA using FactoMineR package in R. I scale the data so all the traits contribute equally. So then after running the PCA, we will run a Scree plot which shows that PCA1 explains 68.6% of variation and the PCA2 explains 13.8% of variation in data. Together, they capture about or over 62% of variation. So I can visualize most difference between varieties in 2D. Next, I check which trait contribute the most. The protein content contributes 28% to the PCA1, followed by the grain yield, seed weight, and plant height. For the PCA2, plant height dominates with 68% contribution. These plots help me understand which trait drives variation among wheat varieties. I also visualize individual wheat varieties. Points are colored by treatment and ellipses show confidence interval. Varieties like Afaq, Sahar, and Galaxy cluster differently. The distance between points shows how similar or different the varieties are in their traits. In the PCA Biplot, points and varieties are arrows show traits. Arrows pointing in the same direction indicate positive correlation. Protein content and the grain yield align with PCA1, confirming their strong influence. I use shapes for the treatments and repel to avoid the label overlap. So finally, I will check the variety contributing the most. Atlas 19, Crimson Wave, Zephyr 8 drive most of the variation. For the PCA2, Omega 9, Triticum Max, and Punjab Gold are the major contributors. This is useful for selecting key varieties for breeding or trait analysis. In summary, PCA shows that protein content, grain yield, and the plant height are the key traits that differentiate wheat varieties in our current data set. Biplots and the contribution plots help me see variety clustering under different treatments, which is essential for crop breeding and trait selection. Thanks for watching Biodecoder. If you like this quick PCA tutorial in R, please like, subscribe, and hit the bell for the more videos.